Hello, Prof. Wan. The paper that I've chosen is whole genome sequencing, Mycobacterium tuberculosis directly from sputum identifies more genetic diversity than sequencing from culture. So first of all, for the introduction, the objective of this research is to identify genetic diversity of Mycobacterium tuberculosis between sputum and cultured samples. The purpose is to study the extent of multi-drug resistant tuberculosis and DRTB as a global infectious disease. It is also to investigate the cause of within patient diversity and heteroresistance. In this study, heterozygous alleles for 12 available paired sequences that were reanalyzed at more than 60-fold mean genome coverage between collected sputum samples from patients with MDR-TB and cultured samples. Next, under methodology, there were 33 patients that enrolled for the study. 2 sputum samples were collected so that one will be inoculated into Mycobacterial growth indicator tube MGIT culture, while the other for direct DNA extraction. For DNA extraction, it is done using mechanical ribolysis, purification using dioscurine lyosine IXT or CTAP, and finally the DNA sequence via NextSec 500 Illumina before being run for read mapping. Variant calling is then conducted to construct phylogenetic tree by calling variants. Single nucleotide polymorphisms were used to combine sequences in multi-alignment FASTA file. Identification of mixed infection is done by screening all samples where samples with 20 or fewer heterozygous SNPs where heterozygous SNPs were left at less than 1.5% of all SNPs were classified as not mixed. Statistical analysis was then performed using PRISM. Mean coverage depth statistics, number of heterozygous alleles, and blast hits of contaminating reads in paired samples were compared using two-tilt Wilcoxon matched pair sign rank test. For the results, we will be discussing patient characteristics and drug susceptibility, genetic diversity, and finally genetic diversity in drug resistance genes. In patient characteristics and drug susceptibility, first-line phenotypic drug susceptibility testing DST has found 20 patients with MDR-TB, where there was only one with rifampicin monoresistance, 2 with isoniazid monoresistance, and the remaining 7 patients with ethambutyrenol resistance. Second-line phenotypic DST was performed for patients with rifampicin resistant or MDR-TB and successfully identified 1 case of kanamycin resistant. In genetic diversity, whole genome sequencing consensus sequence level maximum likelihood phylogenetic tree was constructed to compare consensus sequence from sputum and MGIT culture samples. It was discovered that samples from patient F1066 and F1067 were closely related with only one consensus level SNP separating all 4 consensus sequences. This close genetic linkage may represent direct transmission within a hospital setting, a community transmission chain, or an unlikely cross-contamination during sample collection. Genetic diversity may also occur because of within-host evolution or mixed infection. To prove it, SNP-based barcode was used to identify mixed infection by scanning all heterozygous alleles for a panel of 413 robust phylogenetically informative SNPs that resolves M. tuberculosis into one of 7 lineages and 55 sublineages. The research has successfully found 3 phylogenetic SNPs among the heterozygous alleles. Same heterozygous phylogenetic SNP originated from the same sublineage as other SNPs are present at 100% frequency, with no cases of heterozygous alleles indicating presence of more than 1 or more sublineages. Identified mixed infection in sputum sample from patient F1096 that had 261 heterozygous alleles, which is 10 times greater than in any other sample. Genetic diversity was found in ribosomal RNA genes RRS and RRL, uniquely in sputum samples compared to other genes. Blast hits were run against diverse genes found. From there, at least 30 bases to M. tuberculosis RRS and RRL genes that encode rRNA thumb sequence versus 4 blast hits from MGIT sequences. There are no hits against any other genes with more than 2 sputum heterozygous alleles apart from RPOC, for which there were 3 hits from sputum sequences, but none from MGIT sequences. After excluding samples with mixed infection and removing rRNA gene sequences, frequency of heterozygous alleles in both samples were compared and found that there were 265 heterozygous alleles identified in sputum samples compared to 200 in MGIT samples. Majority of heterozygous alleles were indels and non-synonymous mutations of commonly frameshift mutations compared to missense mutations. Lastly, for genetic diversity in drug resistance, 4 of the 32 patients with single-strain infection had RAVs present as heterozygous alleles in at least 1 gene. Patient F1066 and F1067 were highly related with only 1 consensus SNP SNP difference between all 4 sequences. Both patients had phenotypic high-level isoniazid resistance with no consensus level CAPG or INHA mutation, but had frameshift CAPG mutations present as heterozygous alleles, potentially to cause resistance. Patient F1066 and RF021 had RV1979C and PNCA mutations respectively at low frequency in sputum, which had the potential to confer phenotypic resistance to clofazimine and pyrazinamide. For discussion, whole genome sequencing using DNA from sputum and MGIT culture was performed to compare genetic diversity between methods. All PAT sequences were closely related at consensus level with predicted phenotypic drug susceptibility with over 95% sensitivity and specificity for rifampicin and isoniazid. It was also discovered that rRNA genes have high levels of diversity in sputum samples, believed due to non-microbacterial DNA hybridizing to capture baits may enrich rRNA from other organisms, causing increase in diversity. More diversity was found when sequencing directly from sputum with significantly more unique heterozygous alleles than sequencing from MGIT culture. 2/3 of patients with MDR-TB had been treated for drug-susceptible TB, causing diversity in sputum samples due to drug pressure. The understanding of within-patient M. tuberculosis genetic diversity is increasingly important as detection of rare variants has been shown to improve correlation between phenotypic and genotypic drug resistance profiles. Therefore, we can identify emerging drug-resistant strains. From figure 2, it is evident that there are higher number of heterozygous alleles found in directly sequenced sputum only, light blue, compared to MGIT pink. This once again proves that there are more genetic diversity of M. tuberculosis found in sputum compared to cultured ones. In conclusion, direct sequencing of M. tuberculosis from sputum sample allows higher identification of genetic diversity compared to MGIT cultured samples. Genetic diversity from sputum sample may arise from effects of drug pressure, other microorganisms of the typical of genera, composing the oral flora found from metagenomics assignment and mixed infection, causing within-patient diversity. Patient characterization is important as well to identify the diversity of M. tuberculosis, as some may have heterozygous alleles at sites associated with drug resistance, RAVS. Improvement on RAVS detection over available rapid genetic tests could propel individualized therapy, prevent chances of acquired resistance, and also give insight into bacterial adaptation to the host. That is all. Thank you.